In the last section, we talked about the evidence that the cerebellum corresponds with the behavioral sequence manager required for a complex learning system. So, having established the connection, let's now have a look at the information model for the cerebellum on various levels. This is a high-level view of the information flow that controls motor movements, and as we've discussed, there's two parallel routes, ultimately deriving information from the senses, then going through the brain to muscle control. One is the route from the cortex, through the basal ganglia and thalamus, back to the cortex, and then from the cortex down to the brainstem and the spinal cord. The second is the route we're discussing, from the cortex, through the pontine nuclei to the cerebellum, and down to the brainstem. In the case of cognitive behaviors, they're implemented by release of receptive field detections between cortical areas. So one route goes from the cortex through the basal ganglia and thalamus back to the cortex, and the other route, for frequently used sequences of mental processes, goes from the cortex to the cerebellum and back to the cortex via the thalamus. In both cases, the cerebellar route bypasses the basal ganglia. So the interpretation of cortical receptive fields into recommendations and the selection of the most strongly recommended behavior doesn't occur. And this saves time, the time required for this selection process between each action in the sequence. But on the other hand, it takes away most of the learning capabilities. A change in a sequence, adding or changing any one action, will have to be learned by the basal ganglia route before it can be handed back again to the cerebellum. Once a frequently used sequence has been learned by the basal ganglia route, the sequence is recorded by a group of neurons in the cerebellum, and these groups are called microzones and include associated groups of neurons in the cerebellar cortex, the cerebellar nuclei, and the inferior olive. When the sequence is running under the control of the basal ganglia route, different neurons in the cerebellar nuclei part of the zone establish connections directly onto different neurons in the brainstem that are active at different points in the sequence. At this point, the brainstem neurons are being driven by outputs from the cortex under the control of the basal ganglia. So by this process, different neurons in the cerebellar nuclei acquire the ability to drive the different behaviors in the sequence. The brainstem neuron driving one action at one point in time is activated by the cortical outputs at the appropriate point in the sequence. One of the cerebellar nucleus neurons is active at the same time, so it gets a synaptic contact onto that brainstem neuron, and it can then drive the action directly. A Purkinje neuron in the cerebellar cortex part of the assigned microzone develops a receptive field that corresponds with a very large number of very complex combinations of cortical receptive fields that just happen to be active just before one action in the sequence occurs. This Purkinje is inhibitory and targets all the cerebellar nuclei neurons in its zone except the one corresponding with the same action. So the Purkinje inhibits all the actions in the sequence except one. So once the sequence is initiated, the firing of the Purkinjes in sequence, driven by different mixes of cortical activity, put brakes on all the behaviors in the sequence except the one coming next. So as I said earlier, the Purkinje firings are effectively commands to execute the next behavior in the sequence. And this use of commands is possible because pretty much all the required learning has already been done. The one bit of learning the cerebellum can do is to speed up the sequence, get each action to occur as soon as possible after the previous action. The inferior olive gets information on the performance of an action directly from the retina and the spinal cord without processing through the cortex. So it'll get sensory and body position information very fast. When one action in the sequence occurs, a neuron in the inferior olive part of the zone gets excitatory input from the senses. The inferior olive neuron also gets inhibitory input from the cerebellar nuclei. If the action occurs at just the right moment, these two inputs coincide, and there's no inferior olive output. But if they don't coincide, the inferior olive neuron fires. And an inferior olive neuron targets just the Purkinje corresponding with the same action in the sequence. And the effect of the inferior olive input is to slightly shift the receptive field of the Purkinje, reducing the weights of synapses that were active right at the moment the Purkinje fired, and increasing the weights of synapses active a little bit earlier. Which means the Purkinje's receptive field will in future be detected slightly earlier, meaning that the corresponding action will be initiated a bit earlier, a better match with the exact time at which the action should be initiated. So if we have a closer look at a microsome, it's got a group of about 100 inhibitory Purkinje neurons, at least in a mouse brain, and a group of excitatory cerebellar nuclei neurons. The cerebellar nuclei neurons target muscle control, each neuron activating the brainstem or spinal cord muscles that control one movement. Each Purkinje, or perhaps a group of Purkinjes, corresponds with one action in the sequence and inhibits all the cerebellar nuclei neurons except those that execute the corresponding action. So different Purkinjes inhibit all except one excitatory cerebellar nucleus neurons in the zone. A small group of neurons in the inferior olive, often arranged actually as a column, target those Purkinjes, roughly one inferior olive neuron for each Purkinje. And there's also inhibitory neurons in the cerebellar nuclei that fire at the same time as the excitatory neurons and target the corresponding inferior olive neurons. So if the action doesn't occur at quite the right time, the inferior olive neuron fires, targeting the Purkinje corresponding with the action. At a more detailed level, the receptive field of a Purkinje is defined by one or two synapses from each of over 100,000 cerebellar cortex granule cells. The axon of an inferior olive neuron wraps itself around the dendrite of one Purkinje, making lots of synapses. The weight of a granule cell synapse increases if it fires just before the Purkinje fires. In other words, if it makes a contribution to the Purkinje receptive field detection, much like the synaptic weight increases in pyramidal neurons in the cerebral cortex. But if the inferior olive fires, it decreases the weights of all the recently active granule cell synapses. In other words, shifting the Purkinje receptive field slightly away from its current definition, which will change the exact timing of when it fires. And the inferior olive also seems to increase the weights of any granule cell synapses that got inputs perhaps 100 milliseconds before the Purkinje fired, encouraging the Purkinje to fire a little earlier in the future. A later dopamine input is required to make the changes long-term. In other words, reward feedback on the execution of the sequence as a whole. And at a deeper level of description, the chemical effect of dopamine will be generally similar to what we described earlier for the cortex, acting to influence the actin skeleton to change the long-term size of the synapse. Now, I've tended to focus mostly on motor sequences, but by controlling a sequence of thalamic activations, the cerebellum can activate a sequence of mental processes, again assuming that sequence has already been learned through the basal ganglia and used successfully a number of times. For example, when we're reading, information derived from the sequence of word images has to be processed and combined in the right order, and the cerebellum could record the order. And of course, speech generation involves the execution of sequences of tongue, mouth and throat muscles, and the cerebellum plays a major role in these sequences, again, once they've been learned.